Now let's talk about what happens at the cellular level that leads to all the events downstream. Let's talk about the pathogenesis of multiple myeloma. Now to understand this, let's start off with the basic cell. We have the cell which is designated cell. It is basically postulated as a multiple myeloma cell. It is the cell, the plasma cell that is going to develop multiple myeloma. So that multiple myeloma cell interacts with the bone marrow stem cells with the help of various adhesion molecules. As a result of this interaction, the bone marrow stem cell is stimulated, and with the help of the NF-kappa B proteins, the NF-kappa B leads to the stimulation of the nuclear proteins, and that leads to a release of a large number of cytokines. Specifically, the important cytokines that have been found to be in effect in patients with multiple myeloma have been the interleukin-6, the vascular endothelial growth factor, the VEGF, or the IGF growth factor and cytokine. So as a result of this interaction and as a result of the cytokine production, as a combined effect with the help of cytokine-mediated signaling and the adhesion-mediated signaling, which is a direct interaction of a multiple myeloma cell with the bone marrow stem cell, this triggers a series of events downstream in which, because of the RAF protein production and the JAK-STAT pathway and the phosphatidylinositol 3 pathway, a lot of proteins are produced downstream, and the multiple myeloma cell undergoes rapid transformation. With the help of these various proteins produced by the multiple myeloma cells, it transforms itself into a malignant cell, and it develops all the potentials of a malignant cell. What are those characteristics? The cell becomes autoproliferative. It can proliferate on its own independent of the growth factors. Similarly, it becomes resistant to the drugs, and it becomes anti-apoptotic. And it gives rise to, it can undergo apoptosis on its own without any signaling, or it basically can resist apoptosis of signaling, like, for example, the BCL or the MCL gene. So these are all the characteristics of neoplasia: the self-proliferation, vascular proliferation, drug resistance to the apoptosis signals. The cell cycle growth or proliferation is basically speeded up, and the cells can also migrate out of these their prime locations, which can lead to the metastasis. So the trigger event was an interaction of that multiple myeloma cell with the help of the adhesion molecules, leading to the production of the cytokines, and afterwards downstream stimulation of the various factors leading to proliferation, drug resistance, cell cycle and migration, and all of these characteristics acquired by the cell. This was the basic pathogenesis at the cellular level. Now, what happens when one cell transforms into a malignant cell? Initially, after this cell, we have the development of MGUS, monoclonal gammopathy of undetermined sequence, undetermined significance. In that cell, there are less than 10% of the bone marrow which is covered by the plasma cell. We don't have any other M proteins, and there is no end organ damage. So MGUS can be thought of as a precursor lesion to multiple myeloma. The patients which have developed MGUS, they need to be regularly monitored for over a period of with 6 months or 1 year follow-up visits, because 1% of the patients who have MGUS, they will develop multiple myeloma every year. So the progression rate is 1%. So that's why it is important to have keep these patients under observation, so that if they start developing the signs and symptoms, we can intervene very quickly. Then MGUS can transform into something which we term as smoldering myeloma. It is a type. It is also called an asymptomatic myeloma patient, a myeloma patient without any symptoms. That eventually can lead to various types of multiple myeloma presentation. It can either be intramedullary myeloma, which is a classical multiple myeloma, or it can be someone who develops a rare feature of a multiple myeloma, or a rare type, which is termed as extramedullary myeloma, in which the myeloma cells move out of the bone marrow, and they can basically reside in the lymphoid tissue of the neck or of the pharynx. But these presentations are very rare. The classical myeloma is generally termed as multiple myeloma or the intramedullary myeloma. And then down the stream, this cell line can lead to a rapid efflux of the antibodies heavy chain or antibodies light chain, either IgG, IgA, or the kappa or lambda, or the various combinations of these antibodies. So this was a basic overview of the pathogenesis of multiple myeloma at the cellular level, and then how one cell transforms, leading to the development or the progression of the myeloma.